The next speaker is actually two speakers. So we have a case study presentation, a toxicological evaluation of a new can lining. So our speakers are Dr. Mark Maier, who is now with Shaparian Toxicology. He was formerly with Valspar Corporation and was manager there of the product safety and regulatory biosciences. And this is where he worked on the discovery of safer materials for can linings and ran a program called Safety by Design, looking for non-biologically active components for the polymers. So Dr. Maier's been a toxicologist for more than 30 years. Hard to tell by looking at him. And he's done a lot of work, health risk assessment, risk policy. He's worked in pharmaceutical and medicinal chemistry, as well as now food contact materials. Our second speaker is Dr. Maricel Maffini. She is right now an independent consultant, but has been with a number of organizations in the past, including as a senior scientist with the Natural Resources Defense Council. She was a professor at Tufts and is also still a professor at, I'm going to butcher this, Universidad Nacional del Litoral, which is in Argentina. Hey, I didn't butcher it. Okay. And this is where she got her doctoral degree in biological sciences originally. Okay, she's been doing research on reproductive biology, carcinogenesis, and endocrine disruption for 25 years now.
Good afternoon to everyone here and everyone who is somewhere scattered about the universe. It's going to be really important for you to understand that I am not a Valspar employee at this time, and I will try to divide my personal opinions from anything that might be representative of Valspar's position. But I think that you will see as we go through this presentation that there would be very few things I could say that Valspar personally would object to, because they led the charge to develop these new materials and to reveal the data behind these new materials. And unfortunately, when you lead the charge, you're usually the first one to take fire. So I think Valspar is aware of that, and as am I. So let's get started here, and I'll show you a couple of other things I want to say, too, is that Dr. LeKind, we did not compare notes before she spoke, but everything she said is in some version in this slide. The slides coming up, and the low dose high throughput screening assays that we just heard about are buried in here. So this is really how you bring all of the things that were talked about today together to develop a new material, and we actually did it. This is not what we would like to do. This is what we actually were able to accomplish at Valspar. And I should also position Dr. Maffini in this, and there's a slide in here that will help you understand how our relationships work. But Dr. Maffini was hired by Valspar as a chaperone, not for an endorsement, not for anything, but to help us navigate through the communications and the science to get this done and to get people involved and to get the transparency that we wanted and to get basically tell us how we can prove that we're wrong. We wanted everybody to be able to prove that Valspar was wrong. And if you can't prove we're wrong, then maybe you can conclude that there's a high probability that we're right. And that was the approach that we took, and I'll talk more about that. And I think, do I have to point it somewhere? There we go. So here's the roadmap for what we're going to talk about. The first thing is I'm going to show you the problem that we face. Dr. LeKind was very articulate in explaining the kinds of challenges that you have developing a can lining. It's not you just spray it on and you should be done. You can't just mix it up and throw it on a can. It takes years and years, and this project started probably 7 years ago, and this was before I joined Valspar. I joined Valspar about 5 years ago. The second thing we're going to do is talk about the obstacles, of which there are many, which Dr. LeKind also articulated. We're going to show you our solution, meaning Valspar's solution, and we're going to give some selected results because Dr. Maffini was very intimately involved in some of the more complex animal work that we did, because she's an expert clearly in animal studies that have to do with bisphenols. BPA, she's done a lot of work with that. So she's our expert on animal studies, and so we're going to let her show you that. She's also going to present the data that was done at her alma mater, where she worked, I guess, at Tufts University, where some very, very elegant in vitro work was done by Dr. Soto's lab. So here's what we're talking about. We're talking about can linings, and I wasn't sure how much setup I would have. I had a lot of really great setup today with all the other speakers. But this is what we're talking about. We're talking about a spray lining for cans that's a polymeric substance, and we are talking about beverage and food cans. And all of the complications that go with the different materials that are in those different kinds of cans. And this, to see what we actually came up with, this is the molecule. And I will show you more about that molecule in a while, but that is the polymer that we developed. And you may notice some of the structural features may look familiar, and I'll show you more about that in a minute. So let me just back up a little bit and say how materials were normally selected by both Valspar and their competitors historically. There are a lot of monomers and polymers and uses listed in the the CFR, the Code of Federal Regulations, that you can basically go in, say, I want this molecule and that molecule. It's already been pre-cleared. We don't have to do any clearance for it with the regulatory agencies. We pull them out of the list and we stick them together and we make something out of them. And the lists are available in the EU and the lists are available in the United States. We both have them. Unfortunately, most of those molecules are legacy molecules. They've been around for a long time. There's very little safety data on them. Most of them were grandfathered in. And so there's not a lot known about these molecules. And BPA, by the way, is on the list. So to do this, to do what Valspar wanted to do, is Valspar had to get out of that box, out of that list, and build some new molecules. And so this is pretty much the point where I came in, was how are we going to build new molecules? And new molecules mean you got to have a lot of requirements. Now this is a way you can think about the kinds of problems that you're going to have with developing a new molecule. And Dr. Lekine's, remember the shelf unit with the cans? This is our lame version of that. Okay, hers was much, much better, but it's the same idea. The idea is that BPA-based epoxies would work across all of these different applications, from beverage can ends, which, by the way, is one of the toughest things that you will ever try to coat, because you have to put a rivet through it, you have to shape it, there's a lot of machining that goes on, and that coating has to withstand all of that tooling. So the beverage end, the beverage body, all the food applications that you have, and I won't go through all the details of what these mean, but these are all different food applications with different can constructions. And then finally closures, which are like the pop tops on ketchup bottles and that sort of thing. So what alternatives do you have? Well, if you're not going to use an epoxy, you can use all of these others. You can use acrylics, you can use vinyls, you can use oleos, which are kind of the legacy coating that's been around for a very long time. Polyester 1, polyester 2, there's all these different chemistries that you can use. But each one of those arrows to each one of those other applications takes, first of all, it takes years to come up with that arrow to make it actually work. And second of all, many of them just don't work. So acrylics tend to be relatively fragile. And you can go across and you can find arguments as to why all of those have different problems with different applications. So what are you left with? You're left with an epoxy. So Valspar's thinking, okay, we have to use an epoxy. We have to develop an epoxy because even though we do all of those other things, we've been doing them so long, we still can't get them to work in all applications. So here's the obstacles that you go through when you try to develop a new molecule. And this, by the way, is kind of like reverse drug discovery. I'm involved in pharmaceuticals, and one of the things pharmaceutical companies are trying to do is find molecules that will actually do something, right? We're trying to find molecules that won't do anything physiologically. So that's a little bit of a challenge. It's reverse drug discovery. So when you start building the walls, you will realize that there are just all of these different regulatory, scientific applications, all of these different obstacles that you have got to penetrate before you are ever going to get something to market. And technical performance is usually where you crash and burn. Technical performance is the hardest thing to get because you can come up with really good molecules that have no biological activity, but you can't make a polymer out of them. Or you can come up with really good molecules that react and make great polymers, but oh geez, they're mutagenic, or they're small, reactive, toxic molecules. So it's a real fine line you walk. You've got migration, supply chain, which we're going to talk about later on. Supply chain, as it turns out, is a huge obstacle. And there's a lot of reasons for that. Food regulatory is usually fairly straightforward. Even in the EU, the only problem is it's structural. It's not actually the science. It's the way the politics and the policies have been structured to get these things approved. And then this is the real one that none of us figured we would have to deal with. Is you may know if something is regulated by FDA, like a drug, you are exempt from EPA regulations. You are exempt in the EU from REACH regulations because that's dual regulation and that's not something that's generally acceptable. If you put it inside the can and it's only for food contact, you are exempt from all the EPA regulations because it's regulated by FDA. But we wanted to say, well, okay, Valspar said, well, wait a minute. What about the outside? What if some of this gets on the outside? Then what do you have? So we had to deal with all of the REACH regulations, all of the TSCA regulations, all of the FDA, Health Canada, EU, all of that, which was things we never expected. So it ended up being a huge undertaking. Now, let me talk chemistry a little bit. I hope everybody in the room doesn't freak out over chemistry. I know some of you love this stuff. But, actually, I'm a chemist, so I kind of love it, but I evolved. This is the chemistry of BPA, and there's a lot to be learned from looking at how you make a BPA polymer. The BPA polymer is at the bottom. You start off with a phenol, you react it with acetone, and you end up with bisphenol A, which, by the way, is what the A stands for, is acetone. Okay? You react it and you get BPA. So we all know BPA is estrogenic in vitro, and we're not going to talk about BPA today. We agreed not to do that. But you learn a lot about what you want to not do if you're going to make a substitute. The other thing that's interesting about this, and this is where all the BPA comes from, is that there's two sources. When you make a BPA polymer, you're going to get BPA up at the beginning, and then when you react the BPA to make this intermediate, you put BPA back into the system, again, to react it with this intermediate to put it together. And number two there is where all the BPA comes from that migrates into the food. The first one is pretty much gone. The second one is because you've done that second reaction, you cannot get it all to react. Because remember, we're doing this in big reactors. This is not a round bottom flask in a laboratory. So you have some things don't react. So we learned that. We said, okay, one of the things that would be nice to do would be to get anything that might resemble a bisphenol out of that second step. And then finally, and this is the clincher here, is you've got to have that 4,4' methylene functionality or the polymer won't work. It won't stick. It doesn't have flexibility. It doesn't have the things that BPA epoxies have. And without that, you're just mixing chemicals together. So somehow, Valspar had to figure out how to get that functionality in there, try to get this out of the system in the second step, and build a new polymer that's not estrogenic. That's a tall order. That's a hugely tall order, but successful. Okay? So we'll talk more about how that happens a little bit later. So now, just this background. This is for anyone who hasn't thought about monomers and polymers. And here's where the hazard problem comes in, is if you're going to put monomers together to make polymers, you have a huge tug of war going on here. And that tug of war is between having a monomer that you can actually react together to make a polymer that doesn't have any hazard. Now, how are you going to find a reactive molecule that you can stick together that doesn't also have biological reactivity? That's a big challenge. So there's this tightrope walk that you have to try to figure out something that will react and make a good polymer at the same time has very low or no biological activity. It's not a small challenge. So here's what you'd like to have, is you'd like to have molecules that react together, but like reverse drug discovery, physiologically do absolutely nothing. It's a tall order. And actually, the first time I showed this slide was in El Paso, Texas, to a group there, and we looked out the window, and that's what we saw. Okay, so now what? So what do we do? Well, okay, we have a tall order and a big problem to solve. How is that going to happen? Well, let's look at the chemistry again. This time more in graphic form, without the scary molecules. We need to find some molecules that are not estrogenic. But here's one more problem. This thing This second intermediate, remember the second intermediate from BPA? You use BPA and you react it, you make that intermediate and you put BPA back in and then react it again. This thing has 2 structural alerts for mutagenicity. So now you've got to, you've got to worry about endocrine activity, estrogenicity in particular. Now you've got to worry about mutagenicity. How are you going to solve that problem? It's a huge problem. Okay, so again, bisphenols are the only commercially viable way to get the 4,4' methylene diphenyl functionality that you need. And diglycidyl ethers, these things that are mutagenic, structural alerts for mutagenicity, have to go into the system too. So now you've got this huge conundrum is how are you going to solve these problems? So to go back to how we found a bisphenol, it had to be a bisphenol because that was the only way you could make it. By the way, if you were making a drug and you needed to have that functionality in the drug, you could do all kinds of crazy, goofy chemistry to get it. And you could protect it and deprotect it and add silanes and all kinds of crazy things to build whatever molecule you wanted and create tons of waste, and nobody would care because that's the way drugs work. But this is a commercial product. Okay, this is something that you've got to deal with waste, you've got to deal with scale, you've got to deal with big reactors, and you've got to do it cheap enough or nobody's going to buy it because you could make the perfect molecule here, and if it's like 20 times more costly than what you've been using, you're never going to sell it. So you've got that commercial side as well. So what we decided to do is okay, let's look at a way to screen all these molecules because there's so many molecules. And by the way, our molecule isn't on your list, John. We've missed that one, which is good. Well, I'm sure it will be now. So anyway, one of the things that I was just thinking, okay, look, we could do all this in vitro screening and we could do what Tox21 is doing, but geez, we don't have the resources for one. We don't have the budget for another. There's got to be some kind of easy way to do this. So I took, okay, I'm a pharmacologist. I should be able to figure out something to do to solve this, to find this molecule. So we said, okay, what's with BPA? Why is BPA one of these molecules that has estrogenicity, and as you go down the scale, they start to lose estrogenicity? And oh, by the way, is there one in there somewhere that doesn't have any estrogenicity? Well, I looked at this ortho BPA. So ortho BPA, see the hydroxyl arms are down. I always get criticized for this part, but hydroxyl arms are down. Okay, so it's ortho, right? This thing, BPA, is para. The arms are out to the side. They're very different molecules. They have exactly the same structural components, but they're different molecules. The one on the right is a very, very poor estrogen receptor agonist. The one on the left, BPA, as we all know, is a very good agonist. So what's with this? Well, what's with this, I'll show you this little animation here real quick. Yeah. Oh, am I knocking around? Oh, how about if I take this off? Is it this? Okay, I'm probably moving too. I'll slow down. It's hard for me to do. But so what you're looking at here, I might have to rerun it again. What I found out is that you can actually put these molecules into ChemDraw. And ChemDraw will do this different low temperature equilibrium modeling for you. And it'll move these molecules around and try to get, it pretends they're in the gas phase, and it models them around to see where their lowest energy configuration is. And what I learned was that this is the ortho right here. And that's the para. So BPA would be on the right. This would be the ortho. One of the things that you'll notice about this configuration is that this right here, this angle right here, can actually go on BPA, can actually go flat. And that allows it to fit in the receptor. Whereas this thing, with the ortho configuration, it's kind of stuck in space like this. It can't fit its way into the receptor. I mean, it's a simple, not, electrons of lowest energy and all that. This is a simple, just visual thing. And it turned out that this worked. And we showed that this worked by doing some fairly sophisticated docking modeling by a consultant that did this, and they actually concluded, we showed them good molecules and bad molecules, and they concluded that this C, the C angle, was a really good predictor of what you wanted versus what you didn't want. So the tighter this angle was and the less it moved, that allowed us to look at like hundreds of different molecules in very, very rapid order. So I'm just sitting here looking at molecules move. I had a seizure or two, but when I got done with my seizures, we had a couple of molecules that fell out as being those that didn't do that. So now this This is it. This is where we ended up. I don't have enough time to talk about the whole story about how we got here, but this is where we ended up. Now, you may say, well, what are you doing showing this? What about your competitors? That's part of the story here. That's part of the story, is that Valspar, and I'll show you the man that's behind this in a minute, and he couldn't be here because of a family medical emergency, but my boss, Tom Mallon, said, well, we have to do this differently. If our intellectual property protections are as good as we think they are, as good as they ought to be, then why can't we share this? And that's what we're doing. We're showing you the chemistry. There, I bet you there is not, and this is, and this is my opinion. Again, this is Mark Maier talking, not Valspar, because Valspar didn't say this. I'm going to say it. This level of transparency creates a lot of problems of the supply chain. And the reason it causes problems in the supply chain is they're concerned about Valspar essentially taking the market, okay? But you have to say, well, wait a minute. If we're showing our competitors the kind of chemistry that we did, how is Valspar trying to take the market? Valspar is trying to lead the market. They're out in front. They're going to, and I know there's going to be things that happen because of that. Hopefully not, but it could happen. But Valspar did the right thing, and that's Mark speaking. Now let me go back to the Valspar that says our customers are important to us. Their customers, who are the brand owners, are important to us, and we don't want to make them angry. But here's what Valspar did: is they said they went to all their customers. They went to all their brand owners. They saw this before anybody else saw it. There was no hiding it from them. They had it. They had the opportunity to speak. And this is, again, Mark talking. They had the opportunity to speak. They said, this is great. How are you doing? Okay? So anyway, this is the chemistry. We're showing it to you now. And what we did was we got this molecule, and that's what Maricel is going to talk about, is how we got to that molecule. So now here's the part. Now this is 10 minutes for me, or 10 minutes for everybody. Okay, I really got to get going here. This will go quick. But so why should people believe these molecules are safe from Valspar's perspective? Well, because I said so. Well, that's not really going to work out too well, right? It hasn't worked out, as we all know. So we tried a couple of prescriptive approaches, green screen, type ed. We said, how can we use those to find molecules? Well, keeping in mind that reactants, this would be hydrogen, that would be oxygen, and that would be water. Hydrogen and oxygen make water. Well, water has no resemblance to hydrogen and oxygen, right? So this molecule that we use to make the polymer with has very little to do with the final product. So we said, okay, let's look at green screen. Let's try a green screen. So here's a couple of green screens for you. This is the green screen for one particular molecule. This is a green screen for another molecule. You would look at those and you'd say, oh, geez, they're red. Those aren't going to work out too well. Well, what happens if you react these two together? Well, if this one happens to be sodium, by the way. This one happens to be chlorine. You react them together. The only thing that you get out of this table salt is persistence. It's going to be a little bit red on persistence. It's going to stay around. So we're like, okay, as much as we love green screen, that's not going to work. And I don't want to knock green screen because actually many of the authors of these paradigms have worked with us basically to tell us if we knew what we were doing or not. So I'm not knocking it. It's a hazard-based approach, but it simply wouldn't work because we didn't have a polymer. We were talking about a polymer. So we were working with green screen to try to come up with a paradigm to deal with the polymers, and we have a green screen practitioner to do that. Typehead was another one that we looked at. The only challenge with Typehead is that it really says you can't know anything about endocrine disruption until you get to the animal. So my question is, well, why don't you just start with the animal and skip all that other stuff? It's hard to get out of this circle. Okay? And I'm not knocking Typehead either. It's a really nice menu to choose from. There's a lot of good stuff in there. So the prescriptive approach wasn't going to work, so we did collaboration as the second choice. And this is Tom Mallon. He's the guy that couldn't be here. And these are not Tom's exact words, but this is the philosophy that Tom Mallon has. Is be strong enough to stand alone, be smart enough to know when help is needed, and brave enough to ask for it. And so we did. What you will see here is not an inclusive list. Now, keep in mind, these people, all of these organizations, not one of them is endorsing us. We're not asking for endorsement, but we shared it with them because we wanted them to tell us that we were wrong. We wanted them to tell us what we were doing wrong. We wanted them to challenge us, to find out if we knew what we were doing. And in fact, some of these universities got the material, and we said, here, go work on it. Go work on it. Spend a year working on it. Tell us what you find out. We didn't know a thing for a year. Okay, so we'll get into some more details about that. Maricel will talk a little bit about some of the work that was done at Tufts. This is my last slide, so there will still be time for Dr. Maffini to talk. But these are all the assays that we did. It's a fairly extensive list. We did it all the way from the xylenols to the migrants. Okay? To the migrants, not just the materials that went into it, not just the material that went onto the can, but what comes out of it when you do the migration studies, which Farah helped us a lot with to get done. What happens here? What happens at low dose when you're exposed to these things? We did all these studies, and I'm not going to go through them individually. Dr. Maffini will talk about some of the really cool work that she encouraged us to do that was in vivo. And that's my part. And remember, Valspar didn't say all that. I said some of it, okay?
Okay, so very, very quickly, just to reiterate what Mark was saying, there were two what I call hypotheses that were presented that based on the predictive models and the way the polymer is made, that TMBPF, the monomer, would not have estrogenic activity and it will not be present in the final polymer. So there were ways to assess that, and I will show more details in a minute, in vitro and in vivo testing and migration testing as well. So in the case that these two hypotheses are true, we still have to deal with the fact that the food is going to be contacting the polymer. For that, we also decided to test what was in the polymer, which kind of estrogenic activity may or may not have, and also look at the migrants in particular. So very briefly here, what you see in red is the new approach that Valspar took. And again, most of what I'm going to say is my opinion and my experience, not necessarily Valspar. But these are the new things that were incorporated. So for the monomer to test whether or not it was estrogenic activity, there were guidance type studies, the regulatory type studies performed, including the mammary gland, because it has been shown to be a very sensitive endpoint for estrogenic activity, especially compared to BPA. And then the collaborative testing, studies were done in independent laboratories. There was also migration quantification in the final polymer. And for the polymeric coating, again, it was tested whether or not what comes out from the final product would have estrogenic activity. And that was also done in an independent laboratory. And then we looked at the migrant profile. So very briefly, these are some of the studies that were done. And I am only going to focus briefly on these three. The e-screen study was done at Tufts University, as Mark mentioned, with Dr. Soto. Then the immature, the eutrophic assay that is considered, quote unquote, the gold standard for estrogenic activity in animals was added the mammary gland biology to it, and then the pubertal studies in male and females, also with a mammary gland component. So very, very quickly, the e-screen is based on increased cell proliferation of these cells called MCF7 cells that are breast cancer cells that respond and proliferate in the presence of estrogen. So if you put them in contact with a chemical, if the chemical is estrogenic, the cells will proliferate and be very happy. So I'm going to skip some of the details, but basically this is the dose response curves. This is the positive control, that is 17 beta estradiol, is the typical dose response curve. This is BPA that was also tested as a chemical that has estrogenic activity. And down here, this flat curve is the monomer TMBPF. So At least in the screen, it looked okay, no estrogenic activity. In the uterotrophic assay, again, there are two versions of it. So the one that was run was with the immature females. So these animals didn't reach puberty. And estrogens have an effect on the uterus that in the presence of estrogen, the uterus increases the weight. So that is the endpoint here. And the new endpoint that was added was the biology of the mammary gland. Very briefly here, the exposure is short. It's only 3 days. And that may have something to do with the mammary gland. But again, the monomer was fed to these animals. And as you can see here, the weight of the uterus was not increased, while the ethinyl estradiol treated animals, the weight of the uterus increased as it's supposed to be. Just as a reference, BPA was not tested in this assay, but as a reference, there is a uterotrophic effect or response by BPA. And when the mammary glands were looked at the microscope of the monomer-treated animals and any of the doses, there was no effect, while the positive control animals did have hyperplasias. That is what estrogen does to the mammary gland. So again, so far, no estrogenic activity observed in the animal study. For the pubertal, here there are male and females, and it's basically looking at whether or not puberty is accelerated or delayed in the presence of the chemicals. So the duration is longer, so you have 20 or 30 days, and all these endpoints are looked at. Basically, are the females having a regular cycle? Are they reaching puberty at the time they're supposed to compared to the controls? And this, again, we added an endpoint of mammary gland development because during puberty is when the mammary gland really expands. It grows and invades and reaches the adult size. So it's a period that is susceptible to chemical exposures, and things can go wrong, and we can see that relatively easily. So again, when you look at any of the parameters for puberty, there were no differences between the untreated animals or the monomer-treated animals, no difference in estrous cycle, no difference in the histology of the reproductive organs or even their weight. And when we look at the mammary glands, the nice things about this type of way of looking at the mammary gland is that you can see the pattern of growth. So think of the mammary gland as a tree. So these are the non-exposed animals, and these are the two different doses of TMBPF. And we didn't see any differences in the pattern of growth, in the structures that are naturally present during puberty, like these club-shaped structures here called terminal end buds. The ducts were branching as they are supposed to be. They had these tiny little things called buds that were present across. So again, in this particular pubertal study, we didn't observe any estrogenic effect either in females or in males. Now, when it comes to the coating, the polymeric coating testing, this was mentioned by Dr. Bradley before and Dr. Cooper as well. They were talking about simulants. So a coated metal was exposed to these two different solutions to simulate these two different types of foods. And then what was collected from the exposure of the metal coated with the polymer and extracted from those two solutions was put on an e-screen assay to identify whether or not the component, the final product, was estrogenic. And as you can see here, again, similar type of dose-response curve. There was no estrogenic effect. And this one here, you will ask yourself, what type of curve is that? All these plates were added the same amount of estrogen, 10 to the minus 10 micromolar estrogen, to identify whether the extract had an anti-estrogenic effect. you may think this is an anti-estrogenic effect, but this was actually cytotoxicity. So the cells started to die at these high concentrations of the extract. So if there is time, I can show you some more of that data. But basically, again, the final product, whatever comes out from the polymer, did not have estrogenic effect on the MCF7 cells. So then again, there was an attempt to quantify the TMBPF because it was the second theory, the second hypothesis, and that was done by Farah. And then there was also an analysis of the NIAS profiles. So as you can see here, in both extracts and either directly extracted from the polymer or concentrated, the TMBPF was below the limit of detection of 0.2 parts per billion. And maybe Dr. Bradley can explain this better than me. I'm not a chemist. But there was also this reporting limit or limit of quantification of 0.06 parts per billion, and TMBPF concentration in the extract was also below that. So that looks pretty good. And here is just to give you a sense of what the NIAS look like. These were just the blanks. So you see that even 50% ethanol have some peaks, and here acetic acid. But these are the extracts. And even though you have all these different types of peaks, and we don't know what these molecules are, but we do know that all these things together, when they are added to the E-screen, they didn't have estrogenic activity. So that is all I have to say about the studies. And now, Mark, if you want to finish.
Okay. Is this back on now? Can you hear me, or did it fall off? Or? How's that? There we go. Okay. I'll just hold my head down like this. So here's what we learned. And this isn't what we learned about the science. This is what we learned, we meaning Valspar, learned about the process. And there's a manuscript that's coming out hopefully soon, if we can ever get it through peer review. It's called Evidence of Absence. Safety requires evidence of absence, not absence of evidence, which has virtually no value, which is essentially what you get when you take things out of the book. There's no evidence for or against the safety. Evidence of absence requires thinking partners who are not like you. And a lot of you in this room are those thinking partners that are not like us, that we asked you to disprove that we knew what we were talking about. And hire critics and ask them to prove you're wrong, which is exactly what we were trying to accomplish. So Valspar's position is get your intellectual property in order, make sure it's tight, make sure it's good, because they are actually a business. And once you've done that, freely share your data. And I'll show you how freely sharing they are of their data in just a moment. Finally, be patient, because you're going to have a lot of conflict. When you're leading the parade, you have a lot of conflict, and you take fire first. And be prepared to change your mind, because what you think is going to be the answer may not be the answer. And finally, just to show you the level of transparency that Valspar has decided upon, if you go to Valspar's website, and I'll explain what that means in a minute. If you go to Valspar's website, you will find all of the intact, unredacted data reports from the contract laboratories, from FARA, from the work that was done internally at Valspar. You will find it all right there. And so will the competitors. But that's what intellectual property protection is supposed to be, and that's what patents are for. So what this means is valsparpackaging.com. If you go there, you will find a tab for Valpure, which is the name of the non-BPA platform that Valspar has, which is not just this, it's the polyesters and the acrylics and the other materials. You go to non-BPA epoxy, and you go to study reports. This is the easy way to remember it. You will find intact those reports. You won't be able to download them. You won't be able to copy and paste them, but you can read every one of them. And this is the literal URL if you want to go there. That's transparency. Show me somebody else that's doing that. And that's a Mark statement. So there's nobody else doing that. Okay. Thanks.
Thank you.